Okay, this part of the lecture is going to cover specifically the Illumina next generation sequencing technology, which is called sequence by synthesis, and this is really quite complicated. I'll take you through it bit by bit, but I'll also discuss it in the live session. So the first thing to do is read this document here, which is from Illumina, explains the sequence by synthesis sequencing platform. And then the next thing to do is watch the Illumina sequencing technology video here. Now, this is quite complicated, and there are steps in there that are jumped, and what I'll try to do is explain all the bits that I think you won't understand to make this technology understandable. So we'll start with the PDF document that Illumina have supplied explaining the sequence technology, and this all takes place on what we call an Illumina flow cell. And this technology requires fragmenting the DNA and somehow attaching them to this flow cell and then performing what we call clonal bridge amplification. So if you can imagine taking a piece of, some human DNA, fragmenting it into small pieces, and we actually attach oligos to each end, and those oligos on each end of the fragmented DNA bind to oligos which are pre-populated on this flow cell. Now those oligos come in two different types, and they recognize two different types of oligo on the flow cell. And basically the oligos on the flow cell bind to the oligo linkers that we've added to the end of our fragmented DNA, and that captures the DNA, and then we perform something called cluster generation. And this is, I won't say it's like PCR, but it's a DNA amplification technique that allows up to 1,000 identical copies of a piece of DNA to be created by solid phase amplification. We call that bridge amplification, and I'll show you how that works in this document and in the session, and it's also present on the Illumina video, which you're going to watch next. So we start off by fragmenting our DNA and ligate adapter oligos onto the end, and these adapter oligos on the end are essential for capturing the DNA onto the flow cell, and the flow cell is pre-populated with a dense lawn of capture probes, but these capture probes also act as primers for DNA polymerase, so that when they become captured, DNA polymerase can extend from the capture probe stroke primer and duplicate the DNA that has been captured. So we then get something called bridge amplification, which is helped by having two different sorts of oligos on the glass slide and two different oligos on the end of our piece of DNA, and these can basically bridge over and bind to the other type of oligo, and when they do that, one of them can act as a primer and allow this piece of DNA that has bridged over to be copied. Now this has missed out lots of steps that are present on the video, so it's a good idea to watch the video and keep flipping back to this document because there are lots of steps missing on this. But the end point that we get is effectively two pieces of double-stranded DNA attached to the flow cell, and we basically repeat this process again and again until we get thousands of copies of DNA within a small localized region on the flow cell, and those are all identical in sequence. So after a while we end up with one piece of DNA would have landed here, it would have folded over and met the other type of oligo, so it had been captured by one oligo, it will have folded over and been captured by the other oligo. We add DNA polymerase, and that then creates two pieces of DNA. They can be denatured, and you can repeat the process and get 4 pieces of DNA, and repeat the process until you get 1,000 pieces of DNA, all of identical sequence. So once we get to that point, again, there's a lot of steps missing, but we can make that single-stranded, and then we can start adding in the fluorescent labeled bases and then do base calling for each cluster. Now we're doing this on 100 million clusters on a single flow cell simultaneously, and that is how we get so much data generated. So at this point you've seen the bare basics. It's worth having a look at the video and also having a look at the slides, because I've explained each point sort of point by point. So these slides just show that in a little bit more detail. We've adapted our ligated on our adapters, these pieces of DNA being captured by the lawn of primer stroke probes on the flow cell. These bind one, fold over and bind another one. You add DNA polymerase, and that allows you to copy, to create 2 copies of DNA. You can then denature that, so you end up with two sense and antisense strand of DNA sticking up from the flow cell, and then you allow that process to repeat, and that will give you your generated cluster of 1,000 copies of DNA that can then be sequenced. Once you've got your clusters and you've got 100 million or so of them on a flow cell, you can add in your fluorescent nucleotides, and as the fluorescent nucleotides are incorporated, you can get an emission of a photon of light. And since you've got thousands of bits of DNA, or 1,000 bits of DNA here, you get enough light given off each time G goes in. You get a little yellow flash of light, and that's because you've got 1,000 pieces of DNA all incorporating a G at the same time. And then you can add some C's, and you can see if that particular cluster gives emission in this case blue for C, and then repeat the process for different nucleotides and then build up your sequence. So effectively you're watching flashes of light appear on the flow cell, so 40 million clusters per flow cell. This is a tiny area, so just 20 microns across, has got dozens of clusters all simultaneously being sequenced. If you get two clusters which are too close together, like these here, then they will be ignored from the data. You need the ratio of DNA just right so that you don't get clusters merging together. So data is taken from clusters which are spatially away from other clusters. And then we read the sequence at each individual spot and then put all of that DNA sequence using lots of computing power and align it all. And we do the sequencing both forwards and reverse on the same cluster. So initially we sequence the forward strand, and then we sequence the reverse strand. Okay, so I'm just going to explain the principle again, but using some of the images from the video. So this is showing the lawn of oligo, so you can see two different types of oligo. These are two different sequences which are directly homologous to the two oligos which have been ligated onto our fragmented DNA for sequencing. Okay, so this is the insert of DNA here, and then these are the two oligos which are attached onto the end. And these oligos have several little parts to them. One bit is for binding the oligo or primer that's attached to the glass slide, and then you've got other sections to it. So it's much more complicated than just a capture probe. There are various different parts to it, which we'll see later on in the video. So we can see at this point, one of the oligos on the end of the DNA for sequencing has been captured by one of the oligos on the flow cell. At this point, DNA polymerase has filled in this gap because we've got a little 3' OH here, and DNA polymerase can fill in that gap to duplicate that DNA. The DNA is denatured. Now the important thing here is that what we're doing is washing away the denatured DNA and leaving this new piece of DNA, which is a copy of this one, chemically attached to the flow cell. Now this means that this is now directly chemically attached to the flow cell rather than just being attached via interactions between nucleotides. Okay, so this piece of DNA is chemically attached to the flow cell as we've just seen. What that can do is fold over and come into contact with the other type of oligo, allowing now DNA polymerase to be added. This will bind to a free 3' OH here and copy this piece of DNA, so we now have two pieces of DNA, sense and antisense, both chemically bound to the flow cell. So after denaturing that step, we end up with a forward strand and reverse strand, both of which are chemically attached to the oligos that formed part of that initial oligo lawn. So these are now permanently attached to the flow cell. So that process is repeated, so that you end up with 2, then 4, then 8, then 16 pieces of DNA, and then we can keep on going until we get 1,000 pieces of DNA for each cluster, which looks like this. So this would be one cluster, 1,000 pieces of DNA, both sense and antisense, all chemically attached to the flow cell. So in the video we're at 1 minute 23 in the Illumina video, and at this point we've cleaved off one of those strands. So there's no point sequencing sense and antisense at the same time, because you would get C's and G's being flagged up at the same time, and you wouldn't know whether C or G was on the sense or anti-sense strand. So you cleave off one strand and just leave one single strand attached to the flow cell. What we can now do is add in a sequencing primer, and this is shown as very short. It's not for nucleotides in length, it's just for illustrative purposes. We can add in a sequencing primer, and now DNA polymerase can work its way down this sequence, adding in nucleotides, and as it does, each nucleotide can give out a photon of light as it is incorporated and detected. So we can add our fluorescent nucleotides and see which one's going to be incorporated. Obviously this one's going to be incorporated at this point, and these are not. As it gets incorporated, we get a nice little blue flash there, because this is the location of the cluster where this piece of DNA is. And it's not just this piece of DNA, it's lots of other pieces of DNA, 1,000 other pieces of DNA that are exactly the same sequence. And you just repeat the process until you've built up the full length sequence. So after sequentially adding in nucleotides and allowing DNA polymerase to extend that, we keep getting little flashes from this part here, this part of the flow cell, and we can start accumulating data for what that sequence is. And the sequence is sort of accumulated from that particular sequence. So if we just move that back across, that's the sequence that's been generated coming from that sequence there. Okay, so we have read this piece of DNA, we've generated this piece of DNA, and we've sequenced it, and that's why it's called sequence by synthesis, because as we sequence the DNA, we effectively read the sequence. Now what we can do at this point is denature it, wash it away, and leave us with our original piece of DNA here. And by repeating the bridge amplification that we've already seen, we can duplicate this piece of DNA. The green oligo at the top can fold over and meet up with complementary oligos which are still attached to the flow cell, and then we can duplicate this piece of DNA and then repeat the sequencing in the opposite direction. Okay, so you've seen the bridge amplification step, you've seen the sequence by synthesis. What you should now be able to do is follow the rest of the video and see all of the additional steps that go towards allowing a second read to be performed. And what we really need to then understand is that we have a forward read, then we have a reverse read, and data from those two clusters are then compared. If they are exactly the same, as in direct antisense of each other, then that will be a good clean read. If there's differences between the two, then the data can be discarded. So there are just additional checks that are taken into account. Okay, I think that's probably enough on the Illumina sequencing platform. As long as you understand the concept of fragmenting the DNA, attaching oligos, binding to a flow cell, performing bridge amplification, and getting as far as doing the first read, then that's probably as much as I need you to do. I don't really mind if you don't quite get all of the index read 1 and index read 2 and all of that type of thing, as long as you understand the concept of bridge amplification, clusters, solid phase amplification, and sequence by sequencing, and then understanding that we have to do a very clever computational alignment process to build up the genome, to build up our whole genome. I think that's as much as I would like you to know. Okay, so that's it for part 2. Part 3 is going to be bioinformatics and bioethics issues associated with this type of data.